Dear audience, the primary object for microscopic study of the structure of organs and tissues is a high-quality histological specimen. Along with studying theoretical material, the ability of a student to correctly diagnose microscopic structures is an essential condition for successfully mastering the histology course at a medical university. The widespread implementation of virtual microscopy technologies in teaching histology opens up new opportunities for each student to develop not only theoretical knowledge remotely, but also skills in navigating histological specimens, identifying their microscopic elements, accurately describing the features of their structure and function. This lecture is dedicated to developing such important diagnostic skills related to the topic of connective supporting tissues. The entire diversity of tissues of the internal environment can be represented in the form of a classification. The system of connective and supporting tissues includes hematopoietic tissues, connective tissues proper, and supporting connective tissues. Hematopoietic tissues ensure of blood cell development in the hematopoietic organs, from where mature formed elements enter the bloodstream. Connective tissues proper are the most widely represented in the organism, varying in the ratio of the basic amorphous substance and fibers, as well as the orientation of fibers in the intercellular substance. In this group, fibrous connective tissues and connective tissues with special properties are distinguished. Supporting connective tissues are characterized by a high density of intercellular substance and form the organism's skeleton, hence also called skeletal. They are subdivided into cartilage and bone or osseous tissues, as well as cement and dentin of the tooth. All cellular elements of connective and supporting tissues have a common embryonic origin. They develop from mesenchyme. Mesenchyme is a type of embryonic tissue which fills the spaces between embryo organs. Mesenchyme directly gives rise to blood and most of the body's connective tissues. Mesenchyme comprised of loose elongated stellate cells embedded in the extracellular fluid or matrix. Mesenchymal cells are the source of the development of blood capillaries formed by flat vascular endothelial cells. Capillary lumen contains red blood cells. Blood is a specialized connective tissue consisting of cells or formed elements and fluid extracellular material called plasma. The so-called formed elements circulating in the plasma are erythrocytes or red blood cells, leukocytes or white blood cells, and platelets. Blood cells can be studied histologically in smears prepared by spreading a drop of blood in a thin layer on a microscope slide. In such films, the cells are clearly visible and distinct from one another, facilitating observation of their nuclei and cytoplasmic characteristics. Blood smears are routinely stained with mixtures of acidic eosin and basic methylene blue (ASHA) dyes. Some of these special stains, such as Romanowski and Giemsa, are named after hematologists who introduced their own modifications into the original mixtures. Leukocytes are divided into two major groups, granulocytes and agranulocytes, based on the density of their cytoplasmic granules. Their estimated sizes mentioned here refer to observations in blood smears in which the cells are spread and appear slightly larger than they are in the circulation. Granulocytes possess two major types of abundant cytoplasmic granules, lysosomes, often called azurophilic granules, and specific granules that bind neutral, basic, or acidic stains and have specific functions. According to the coloring of the granules, all granulocytes divide into neutrophils, eosinophils, and basophils. Mature granulocytes have segmented nuclei. Agranulocytes lack specific granules, but do contain some azurophilic granules or lysosomes. Their nuclei are not segmented, spherical, or bean-shaped. This group includes the lymphocytes and monocytes. An adult blood smear. Erythrocyte is oxyphil, a nuclear cell with lighter center. These are factually cell-derived structures. Segmented neutrophilic granulocyte or neutrophil is orbed cell with lobed nucleus consisting from 2 to 5 segments joined together by hair-like filaments. Its cytoplasm contains very small lilac granules. Small lymphocyte is orbed cell with a large round nucleus which occupies nearly all body of the cell and thin rim of basophilic cytoplasm. Eosinophilic granulocyte or eosinophil is orbed cell with nucleus which often consists of 2 segments. Large eosinophilic granules are located in cytoplasm. Blood platelets or thrombocytes are a cluster of small and slightly basophilic corpuscles. Every blood plate consists of a light transparent outer part named hyaloma and a central colored part named granulomere containing basophilic granules. Non-segmented neutrophilic granulocyte is orbed cell with small lilac granules in cytoplasm. Its nucleus is not segmented, but has a band or rod-like shape vary. Monocyte is large orbed cell with bean-shaped nucleus and cytoplasm containing poorly manifested non-specific slightly basophilic granules. Basophilic granulocyte or basophil is orbed cell with nucleus of irregular form and cytoplasm containing large basophilic granules. Medium lymphocyte is orbed cell with a large round nucleus and basophilic cytoplasm, which may contain nonspecific aterophilic granules. Connective tissue proper is widespread in the body. It is divided into two general types, fibrous connective tissues and special connective tissues. Fibrous tissues broadly classified as loose or dense, terms that refer to the amount of collagen present. Loose connective tissue is common, forming a layer beneath the epithelial lining of many organs and filling the spaces between fibers of muscle and nerve. Also called areolar tissue, the loose connective tissue typically contains cells, fibers, and ground substance in roughly equal parts. Dense connective tissue has similar components as loose connective tissue, but with fewer cells, mostly fibrocytes, and a clear predominance of bundled collagen fibers over ground substance. Depending on the orientation of collagen bundles, this tissue is subdivided into dense regular and dense irregular connective tissues. Loose connective tissue. The specimen is a thin film of dermal connective tissue after fixation and extension followed by staining by iron hematoxylin on the slide. The numerous cells of different types are located in the amorphous ground substance with loosely arranged collagen and elastic fibers. Fibroblast is a large irregularly shaped process cell with blurred boundaries and a light nucleus. Collagen fibers are tortuous cords running in different directions, forming bundles of variable thickness. Elastic fibers are thinner than collagen fibers. They do not form bundles and anastomosis. The ground or amorphous substance stains weakly basophilic and fills the spaces between cells and fibers. Macrophage or histiocyte is a cell with clear contours, has an uneven shape, granular cytoplasm, and a small dense nucleus. The skin contains epithelial, epidermis, and connective tissue, dermis and hypodermis, components. Loose connective tissue lies beneath the epidermis and forms the papillary layer. Dense fibrous irregular connective tissue forms the reticular layer. The main component of the hypodermis is white adipose tissue organized into lobules. Loose fibrous connective tissue is rich in cellular elements, contains thin collagen fibers and blood capillaries. Dense fibrous unformed connective tissue is represented by thick bundles of collagen fibers oriented in different directions. Cellular elements, mainly fibrocytes, are few in number. The tough three-dimensional collagen network provides resistance to stress from all directions. The dense regular connective tissue consists mainly of not numerous fibrocytes and densely packed bundles of collagen fibers forming parallel structures of great tensile strength. The tendon is formed by numerous longitudinally oriented thick collagen fibers surrounded by fibrocytes. Interlayers of loose connective tissue within tendons denoted as endotendon. Each collagen fiber separated by fibrocytes is designated as a bundle of first order. Endotendon is formed by loose connective tissue and contains blood vessels that supply the tendon. Groups of first-order bundles surrounded by endotendon form second-order bundles. The group of special connective tissues includes adipose, reticular, and mucous tissues. The predominant cells of adipose tissue are adipocytes. There are two types of adipocytes and accordingly, two types of adipose tissue, white and brown. In humans, brown adipose tissue is found in newborns and is reduced by the end of the first year of life, after which white adipose tissue remains the only type of adipose tissue. Reticular tissue consists of stellate reticular cells, a network of thin argyrophilic fibrils. It provides a microenvironment for the development of hematopoietic cells within the bone marrow, lymph nodes, and spleen. Mucous or gelatinous connective tissue is found in humans only during the intrauterine period and is the main tissue component of the fetal umbilical cord. White adipose tissue of skin hypodermis. The main component of the hypodermis is white adipose tissue organized into lobules. The lobules are separated by septa of loose connective tissue conducting blood vessels and nerves. Adipocyte is a large round cell containing a large fat drop. Their cytoplasm is compressed into a thin rim and the nucleus bulges out on one side. After standard histological processing, the fat cells appear empty because the lipid was dissolved during slide preparation. Thin collagenic fibers can be seen between the cells. Reticular tissue. Reticular tissue is represented by a network of loosely arranged branched reticular cells, an intercellular substance containing reticular fibers and amorphous substance. Reticular fibers are not detected by standard staining methods and are visible only when impregnated with silver salts. Reticular cells, with the help of processes, form a three-dimensional network. Developing blood cells are located in the spaces between the reticular cells. Reticular fibers are revealed when impregnated with silver salts and have the properties of argyrophilia. Supporting connective tissues are characterized by a high density of intercellular substance and form the organism's skeleton, hence also called skeletal. They are subdivided into three types of cartilaginous tissue: hyaline, elastic, and fibrous. Two types of bone tissue, woven or reticulofibrous, and lamellar, as well as cement and dentin of the tooth. Cartilage is a tough, durable form of supporting connective tissue characterized by an extracellular matrix with a high concentration of glycosaminoglycans and proteolycans interacting with collagen and elastic fibers. The extracellular matrix predominates and determines the cartilage mechanical properties, hardness, flexibility, and resilience. Cartilage has no blood vessels of its own and exhibits a low metabolic activity. Its nutrition is provided by vessels of neighboring connective tissues. Hyaline cartilage of the rabbit rib. The cartilage is covered on the outside by perichondrium. The outer layer of the perichondrium is represented by fibrous connective tissue with blood vessels. The inner layer of the perichondrium is formed mainly by poorly differentiated cells called prechondroblasts. Beneath the perichondrium, in the lacunae of the intercellular substance, chondroblasts, young spindle-shaped cartilage cells orientated parallel to the cartilage surface, located singly. Deeper in the lacunae there are chondrocytes, mature cartilage cells of oval or round shape. Chondrocytes located in a common lacuna form isogenic aggregates of cells that are descendants of a single chondroblast. Accumulations of single chondrocytes and their isogenic aggregates or cartilaginous territories are surrounded by a basophilic territorial cell matrix with blurred boundaries. Interstitial matrix is the oldest areas of intercellular matter, stained weakly basophilically. Elastic cartilage of the auricle. Elastic cartilage is covered by perichondrium. The outer layer of the perichondrium is represented by fibrous connective tissue with blood vessels. The inner, chondrogenic, layer of the perichondrium is formed by poorly differentiated cells, prechondroblasts and chondroblasts. Chondrocytes have a round or elongated shape, located in lacunae singly or form small isogenic aggregates. In the intercellular matrix, when stained with ossin, a dense network of elastic fibers is revealed. Elastic fibers are thicker and are located more densely in the deep areas of the cartilage. Bone is a supporting connective tissue consisting of calcified extracellular material, the bone matrix, which is produced by specialized cells of the fibroblastic lineage, osteoblasts and osteocytes. Bone tissue is most rigid and hardest of all supporting tissues. It is adapted to resistance, tension and compression. Bone tissues are classified into woven and lamellar bone. Woven bone is the principal bone tissue type in the fetus. Later, woven bone is replaced by lamellar bone. In the matrix of woven bone, collagen fibers are not well ordered. The tissue is poorly mineralized, resulting in low mechanical strength. Lamellar bone is the main tissue of the bone skeleton in adults. The matrix is mineralized and made up of bone lamellae, which contain parallel collagen, ossin, fibers to form complex spatial structures inside the bones. The lamellae are arranged in parallel sheets or concentrically around the central canal, forming the structural units of bone, osteons. The process of bone development is referred to by the term osteogenesis. Lamellar bone tissue, cross-section of tubular bone. The structure of a tubular bone fragment on a transverse section of the diaphysis. The periosteum covers the bone from the outside. A layer of external circumferential lamellae covers the outer surface of the bone. The layer of internal circumferential lamellae is located on the border with the bone marrow cavity. The osteon layer forms the bulk of the compact substance of lamellar bone. The outer layer of the periosteum is formed by dense fibrous connective tissue, the fibers of which pass into the areas of attachment of ligaments and muscles. The deep inner layer of the periosteum is represented by loose fibrous connective tissue with osteogenic cells. Osteon is structural and functional unit of lamellar bone. Each osteon in cross-section has the appearance of a cylinder formed by bone lamellae concentrically located around the osteonic canal. The osteonic canal is lined by osteogenic cells of the periosteum and contains a small blood vessel. There are bone lacunae between the bone lamellae, which contain osteocyte bodies. Osteocyte processes are located in numerous bone canaliculi penetrating the lamellae at right angles. The spaces between the osteons are filled with interstitial lamellae, which are remnants of older generation osteons. Bone development or osteogenesis occurs by one of two processes. Intramembranous ossification, in which osteoblasts differentiate directly from mesenchyme and begin secreting osteoid. Endochondral ossification, in which a pre-existing matrix of hyaline cartilage is eroded and invaded by osteoblasts, which then begin osteoid production. Intramembranous or direct ossification, development of woven bone from mesenchyme. Woven bone tissue is represented by intensely eosin-stained bone trabeculae between which there are skeletogenic islands of mesenchyme. Mesenchyme consists of loosely located cells with large nuclei and thin processes with which they come into contact and form a three-dimensional network. Skeletogenic islands of mesenchyme contain blood vessels filled with erythrocytes, the wall of which is formed by a single layer of endothelial cells. Some mesenchymal cells divide mitotically. Osteoblasts are polygonal cells with a large nucleus and basophilic cytoplasm, arranged in a single layer around the periphery of developing bone trabeculae. Osteocytes are localized within the mineralized osteoid of the bone beam in narrow bone lacunae. Osteoclasts are large multinucleated cells located on the surface of the bone trabecula in resorption bays singly or in small groups. The plasma lemma of the osteoclast forms numerous folds signed as ruffle border. The intercellular matrix of the bone trabecula is oxyphilic and homogeneous because collagen fibers are not arranged in an orderly manner and are not visible on the preparation. Endochondral or indirect ossification, replacement of cartilage with woven bone. Stage of perichondral and endochondral ossification in diaphysis. Bone epiphyses consist of hyaline cartilage on the surface of which the perichondrium is located. The diaphysis is externally limited by the periosteum and a perichondral bone cuff around the cartilage model. Progenitor cells of the inner layer of the perichondrium differentiate into osteoblasts, from which the periosteum is formed. The formation of the perichondral bone cuff follows the mechanism of direct osteogenesis due to the differentiation of osteoblasts producing bone intercellular matrix. Foci of endochondral ossification are located in the central part of the diaphysis. Endochondral bone formation occurs from osteogenic cells surrounding blood vessels that grow deep from the periosteum. Developing foci of endochondral ossification replace areas of calcified cartilage. Between the areas of extravascular endochondral ossification, there are foci of secondary hematopoiesis, red bone marrow formation. In the cartilaginous areas of the epiphysis, there are 4 zones not sharply delimited from each other. Zone of unchanged cartilage or reserve zone. Zone of cartilaginous columns or proliferative zone contains columns of chondrocytes actively dividing and producing intercellular substance. Zone of vesicular cartilage consisting of large dying cells. Zone of calcified cartilage adjacent to the endochondral bone of the diaphysis. Dear students, the technologies of navigating through virtual histological preparations open up new wonderful opportunities to gain solid knowledge in histology. I have no doubt that the combination of the classical teaching model with the latest achievements in digital technologies will allow you to successfully master our fundamental discipline and develop the professional competencies necessary for a future doctor. Stay with us for our next meetings on the HistoNavigator channel.